The second presentation is by Salit Lazare, and Salit is a researcher. She's associated with the Galat Research Center in Israel, which is in southern Israel. She also has a lot of work experience in flowering plants, especially liliums. And she's also currently the head of innovation at RCK Ruhama, and she's doing research on micropropagation and similar things to that. So, but today she's going to talk to us about research that she conducted with our known dog on salinity of avocados. Salit.
Yes, I'm just trying to share my screen. Do you see that? I hope so.
Yes, we do.
So, as Marilu said, most of my talk will deal with some of my postdoc research, which was done in the lab of Arnon Dag. And I'll speak about the rootstock dependent response of Hass avocado to salt stress. But before I'll begin with the research, we see a wide range of plant responses to salinity exposure, and we see that in different levels. We see an increase of bad things in the molecular level, lipid oxidation, protein denaturation, gene methylation, and others. In the cellular level, we see ionic imbalance, degradation of the cell wall, rigidification of membranes, and other bad things. And when we speak about the whole plant, we see a decrease in physiology parameters, photosynthesis, water uptake, nutrient uptake, and of course in the development, vegetative and reproductive growth, and of course the yield. So we focused on the rootstock and scion relationship regarding the responses to salinity. And this is our experimental orchard in Gilat. We have here 600 trees, about 20 different rootstocks. And you can see here that we have 100 plots. So each rootstock had 25 repetitions in 5 plots. And in each plot, you can see we have 5 trees of Hass grafted on the same scion and 1 tree of Ettinger as a pollinizer on the same rootstock. The rootstocks are of different races. Most of them are West Indian, some are Mexican and Guatemalan. Most of them are clonal propagated, VC, and some are seedlings. And most of them are from Ben Yaakov collection, and some are from different collections in Israel and outside of Israel. So before we began the salinity exposure for several years, the orchard was fertigated with fresh water. And you can see that under fresh water, we have significant differences between the rootstocks regarding the mineral content of the leaves. We see here chloride, magnesium, calcium, and manganese that are affected by the rootstock. And when we tried to group them, we saw that the Mexican rootstocks had significantly higher levels of chlorides in the leaves compared with the West Indian, and the West Indians had higher levels of magnesium and manganese. Along the talk, I will say leaves regarding the rootstocks, although the leaves are of Hass. Okay, so it's just for I declare it now, so I'll say roots of this rootstock and leaves of that rootstock, but all the leaves are of Hass. So on March 2019, we began the experiment. And we wanted to mimic the treated wastewater of Israel. As David told you, most of the agriculture in Israel is using treated wastewater, and for over 30 years, the avocado orchards in Israel are fertigated with this water. So in Gilat, we didn't have treated wastewater. We had only fresh water, but we tried to mimic that. So we added the NaCl, sodium chloride, and we tried to reach 280 milligrams chloride per liter, as the INBA regulations declare that this is the right chloride level for treated wastewater. And we tried to reach EC of 1.4, 1.5. And in our tap water, the fresh water, we have about 30 to 40 ppm of chloride. So we tried to reach 280. And you can see that most of the time we had an EC of about 1.4, 1.5. But sometimes we have these jumps, ups and downs. But when we checked, we analyzed the water, you can see that most of the time the chloride level was the right one, although the EC changed. And that's because the tap water changed in their content. And here you can see we have 1.8. We were really afraid. We thought there was some problem with the pump, and we analyzed the water and we saw that the chlorides were fine, but there was a jump there in the what it was, magnesium and calcium. So most of the time the EC was fine. So after 2 years of salinity exposure, the average growth of the trees in all the orchard was about 17%, the relative change in the trunk circumference. But you can see that some of the rootstocks had lower levels of growth. VC 840 grew only in 11%, DUSA only in 14%, and some of the rootstocks performed better. VC 140 21%, and VC 152 26%. This is regarding the vegetative growth. I'll show you here several stress indicators. Some of them need to be measured by tools, by machines, and some of them you can measure by hands. And it's important to try and measure the stress of your trees, even though not all the time we have specific tools for that. So here we'll see a survey, visual survey of salinity damages. Each box here in this map represents a tree. The black here are dead trees, and not because of the salinity long ago. And all the trees were rated from 0 to 3, when 0 means we had no salinity damages in the leaves. 1 means we had low level of salinity damage. I know this is not low level, but you can see that the others are worse. So this is 1. 2 means heavier damage, and 3 means we had defoliation of the leaves. So each rootstock, you can see in these pie graphs, pie diagrams, was different. Some of the rootstocks performed better. You see here again, VC 140. All the trees were rated 0, means they had no visual damage. And others had much more damage. VC 840, all the trees had salinity damages. Another indicator was by remote sensing, drones that we sent above the orchard. There are several stress indicators that can be tested by remote sensing. The usual one is called NDVI, but we found that NDVI wasn't suitable for our avocado orchards because the chlorophyll saturation of the leaves. So this one, NDRE, was better. And you can see here that we had significant differences between the rootstocks, the stress indicators of NDLE. You can see that the ones that suffered the most were DUSA, Nachlat 3, and VC840. Remember this name. We'll find it again and again. And then we tested photosynthesis indices. And you can see here stomatal conductance and CO2 assimilation. And the rootstocks that performed the best were VC140, VC152, VC159, here and here. And another indicator was the leaf osmolality, meaning how much osmolytes we have in the leaves. And the one that had the highest level of osmolytes was, again, VC840, that we saw that suffered the most. And we saw in the mineral content analysis, it had the highest level of chlorides in the leaves. And VC152 had the lowest leaf osmolality. Then we took the leaves to the microscope. This is the abaxial side, the lower side of the leaves. These pores are the stomata. Here you can see it closer. And when we tested these leaves under stereoscope, we found that in the rootstocks that suffered more from salinity, there were higher levels of trichomes, these hairs that can be seen by eye, but in the stereoscope they are seen better. You see that in VC840 we had higher density of the trichomes, and in VC152 we hardly see them. Now the density of trichomes is a known salinity indicator in other plants, not many, but in some. And we found out that in avocado as well, it can be an indicator for a tree that suffers from salt exposure. And here you see the graph. The trichome density is highest in this rootstock that suffered the most in all the previous indicators as well. And now if we look on the yield, okay, this is what we're looking for. And the fruit, again, we see that we have very, very obvious differences between the rootstocks. Some of them, this is a fruit per tree. Some of them have higher yields and some have lowers. And we took all the stress indicators that we measured, not all of them I showed you, leaf area index and others, and we rated all the rootstocks from 1 to 20, when 1 means that the rootstock suffered the least from the salinity, and 20 means it suffered the most. So if we're looking on the leaf chloride in 2020, then the rootstock that had the lowest level of chloride in the leaves is VC152. And the one that had the highest level of chloride in the leaf was DUSA. Okay, so regarding each of these indicators, we rated the rootstocks. And this is the final score. We rated all of them. And you can see that the most susceptible rootstocks were VC840, DUSA, VC842, 802, sorry. And the ones that were the most tolerant were VC159, VC152, and VC140. Okay, so this is the final score. in these conditions, okay, on the EC of 1.4 or 1.5. And in under our conditions in the south of Israel. Now, I want to show you something interesting. Dusa, that we found to be one of the most susceptible to salinity, was tested in another research that was that was published in 2018. And you can see here, these are rootstocks in California. And Dusa is one of the most tolerant ones, okay, to salinity. And we guess that the reason for the difference between our results and these ones are because our rootstocks are from the Israeli collection, and the Israeli collection contains rootstocks that after 30 years of irrigation in treated wastewater, we know what they behave like. So the beginning point was different, and it's interesting to see the differences. Anyway, we wanted then to understand why some rootstocks are more susceptible and others are more tolerant. So we chose 2 rootstocks, the one that was that suffered the most, VC840, and the one that was the most tolerant, VC152. And we tested their ionome, the mineral content, the metabolites, and the lipids in their roots and in their leaves. And you can see that after 8 weeks of salinity exposure, the rootstock that suffered the most from the salinity had higher levels, significantly higher levels of most of the minerals in the leaves compared with the tolerant one. And the tolerant one had higher levels of all the minerals in its roots. We had no deficiencies in the leaves of the tolerant one, but we didn't like this result. We thought that maybe we have here some kind of incompatibility between the rootstock and the scion, and that's why we have here more minerals than in the scion. And in order to test that, we drilled into the trunk in the scion part and in the rootstock trunk. We weren't very kind to the trees, I admit. We drilled there and we analyzed the mineral concentration. And our hypothesis was that if there is an incompatibility between the rootstock and the scion, we will have higher levels of the minerals here at the trunk below the grafting. And that was not the result. You can see that regarding most of the minerals, there was no excessive levels in the rootstock trunk, but there was higher levels of sodium and copper and iron compared with the scion trunk. This is in the VC152, and in VC, the other one, let's call it the tolerant one and the susceptible. Okay, so in the susceptible one, we hardly see any difference between the 2 parts of the trunk. But if we're looking on the yellow marks, these are the differences between the rootstocks. So we see that we have significantly higher levels of some of the rootstocks, of some of the minerals in the tolerant rootstock. But I want to show you specifically the sodium. We see that in the tolerant rootstock, we had higher levels of sodium in the rootstock part of the trunk, 4 times more than in the scion part of the trunk. And the susceptible rootstock had significantly higher levels of sodium. And you can see that we have 5 times more sodium in the rootstock trunk compared with the tolerant rootstock, and about 26 times more in the scion trunk. So the susceptible rootstock is susceptible, and it delivers the sodium up, and it holds it in the trunk and in the leaves. We'll see it later on. So we saw that not only the salt minerals, not only the sodium and the chloride were held in the scion parts of the tree. We did the metabolomics, and we found out that carbohydrates were also higher significantly in the leaves of the susceptible rootstock compared with the tolerant one. And when we saw that, we wondered whether this rootstock does more photosynthesis. How come it has more sugars, carbohydrates in the leaves? So we tested the photosynthesis. You see that the tolerant rootstock had higher levels of chlorophyll in the leaves, and the photosynthesis indices were higher as well. You can see here assimilation, transpiration, and stomatal conductance. So the tolerant rootstock does more photosynthesis. So where are all the carbohydrates? If we're looking on the leaves roots proportion of the sugar intensity, we see that without salinity, there is no significant difference between the rootstocks. Fructose, glucose, and sucrose are higher in the leaves compared with the roots. Okay, 4 times more fructose, for instance, in the leaves compared with the roots. But monoheptulose and persitol, which are the major sugars in avocado, are quite even between the leaves and the roots. The proportion is close to 1. This is without salinity. But after 8 weeks of salinity exposure, 52 days or 8 weeks, about 8 weeks, doesn't matter, we see that in the tolerant rootstock, there was no significant difference in the fructose and in the sucrose and in the persitol. But we see that in the glucose, we have still more carbohydrates in the leaves compared with the roots. But before the salinity, we had 36 times more sugar in the leaves compared with the roots. And after the salinity exposure, we have 4.5 times more, okay? So the proportion decreased. And in the monoheptulose, we see that we have higher levels of this sugar in the roots compared with the leaves. Okay, in the susceptible rootstock, we see that we have higher levels now of the sugars in the leaves compared with the roots. And even in persitol, it's not even anymore. We have more in the leaves. So the proportion changed. If we're looking on the lipids, we see that the storage lipids in the leaves, which are, they have the same function as the sugars, we see the same picture like the sugars. The chloroplast lipids are higher in the tolerant rootstock, and the lipid membranes are interesting. In the first slide, I showed you that one of the salinity responses of plants to one of the stress indicators to salinity in plants is membrane rigidification. It means that the metabolism stops. And here in the tolerant rootstock, we see exactly the opposite. We had an increase in the unsaturated forms and a decrease in the saturated ones, meaning the membranes became more liquid. So the plant could do more, the metabolism was high. Okay, this is exactly the opposite of what we were looking for when we speak about salinity exposure. Regarding the lipids in the roots, we see what we could thought, what we could think. The lipid storage, the storage lipids, the plastid lipids, and the membrane lipids were higher in the tolerant rootstock compared with the susceptible one. So to summarize, if we have these two rootstocks and we expose both of them to high salt water, we see that the susceptible rootstock transport the salt minerals, the toxic minerals, up to the scion, while the tolerant one holds them in the roots. In order to hold them in the roots, to be selective, it must get energy. So the scion somehow senses this need of the roots. And we saw that we had more chlorophyll and more photosynthesis, and all the sugars, the TCA cycle elements, the lipids, not all, but much of them is down here in the roots. It is transported from the scion to the rootstock in order to give it energy. And we see that although so much energy is going down to the roots, we still have good vegetative growth and good yield, meaning that we have no problem with this giving up of some of the sugars and the energy. So we see that we have a relationship between the rootstock and the scion, and it's reciprocal. And we must consider the affinity between the rootstock and the scion when we speak about stress. We were talking about Hass, maybe in other scions, other rootstocks will do better. It is very, very important to select the right rootstock. And this is a very powerful tool when we need to mitigate salinity damage or other damages when we're using low quality water. That's it. Here you have 3 of our papers if you want to read more. They were published already, and I'll be happy to hear questions. Thank you.
Thank you, Salit. That was very interesting. And just to remind you again, those 3 publications are already posted on avocadosource.com. And these PowerPoint presentations in PDF format will also be there hopefully within the next week, as soon as David and Salit provide me their presentation.